So I'm going to talk today about the effects of the Y chromosome on autoimmune and infectious diseases as arose as our studies out of the interest of our studies in the sexually dimorphic traits in autoimmune and immune responses over the years. The underlying premises or hypothesis that is associated with many of these, again, I'm an evolutionist, so you have to put things in an evolutionary context. And in the evolutionary context of infectious diseases and autoimmune disease and survival is simply the evolutionary endpoint of being able to reproduce and pass on your genes. So things that happen past that point in time are actually considered evolutionarily not broken, because things that occur before that point in time is where evolutionary selective pressures were impacted the outcome of the phenotypes of interest. Along this line, it's very clear that more males die than females each year. This has been hypothesized to be due to a number of different mechanisms: behavior, parental nurturing, and disease burden. And with respect to disease burden, there are males of many species ranging from humans to insects are more susceptible to parasitic, fungal, bacterial, and viral infections, as Sauber pointed out. And the primary hypothesis which has been put forth to account for this is called the immunocompetence handicap paradigm, which posits a trade-off between the role of testosterone and driving secondary sex characteristics and suppressing the immune response. However, it's very clear, again, if you look at this evolutionarily, that the immunosuppressive effects of testosterone are age and species specific. What you see in males and human males is very age dependent and will be very different in terms of age dependency in different species. Moreover, the sexual dimorphism that's observed in the species in which testosterone is not the sex hormone. For example, you see this same difference, the sex difference between males and females in species of organisms which in which testosterone or estrogen are not the sex hormones driving differentiation and sexual differentiation. So basically, this model fails to explain the evolutionary ubiquity of the phenomenon that is seen. And if you look now, if you take a lesson from evolutionary lesson from Drosophila, there's actually more to the Y chromosome than junk and sex. And that's based on studies from a number of laboratories looking at the looking at the effects of Y chromosome genetic variation in Drosophila. The very first phenotypes observed were behavioral phenotypes. There's a profound effect of different Y chromosomes of different origins in terms of their genetic evolution on behavioral phenotypes in flies. And it also alters the state of the heterochromatin and the euchromatin within cells. And it was shown very elegantly by Bernardo a few years ago that the genetic variation in Drosophila chromosomes epigenetically regulates the expression of autosomal and X chromosome encoded immune response genes. And therefore, this was attributed to the number of functional copies of variation in multicopy ribosomal genes. Now you have to remember that the sex chromosomes, as they evolved, became separated from each other in terms of sex determining aspects, and then genes that were present on both chromosomes then were no longer free to recombine. And then oftentimes duplications and multiple duplications and variations occurred in those kinds of genes, then that there was no ability to rebalance in terms of recombinational events. So you have X chromosomes and Y chromosomes, which will have evolutional orthologs, but will have huge differences in copy number variation between them. And there you have a potential for huge, what is called intergenomic XY chromosome conflict. And we'll touch on that slightly in a second as a possible mechanism to underlie many of the sex differences that we see independent without having to invoke sex hormone influences. The role of the Y chromosome of genetic variation of the Y chromosome in diseases that are primarily of immune interest we first described that EA severity in both males and female mice was influenced by the origin of the Y chromosome. Later we showed that the basal level of invariant NKT cells was also influenced by the Y chromosome. There was a paper published in 2013 in which was a spontaneous mutation that occurred in a strain of mice with a Y chromosome in which there were no NKT cells and B cells, and this was not within the region of the Y chromosome that was encoding genes that are normally expressed and involved in sexual differentiation. It was in the more heterochromatic regions, again supporting the notion that heterochromatic regions of the X and Y chromosomes may have profound epigenetic influences on driving immune systems and development of immune responses. Importantly, Y chromosome polymorphism has also been reported to be important in cardiomyocyte size, and in this case there was an interaction with neonatal testosterone in driving cardiomyocyte development. We published in 2009 that the sexual dimorphism that Rhonda referred to in the SJL mouse is actually a developmental effect. In terms of, you look at the age of mice, that sexual dimorphism changes as a function of age in male and female SJL mice, and that if you replace the Y chromosome in the males, you get rid of that sexual development. So the actual sexual dimorphism is not a function of the female, it's actually a function of the male mice and their response to EAE, which is Y chromosome dependent. And then importantly, in human disease, there's a haplotype group, one variation that was shown recently to be linked to coronary artery disease in humans. So, based on that, we set out to test the hypothesis that natural genetic variation in mammalian chromosome, Y chromosomes, may influence susceptibility to autoimmune diseases and infectious diseases, as in Drosophila, by epigenetically regulating genome-wide gene expression in somatic cells relevant to disease pathogenesis. And in order to do that experiment, we developed, or took advantage of, a panel of chromosome substitution strains, or consomic strains, in which the black 6 background, C57 black 6 mice, were mated with males of a particular donor strain of origin, of interest, and then those males were continually back-crossed to female black 6 mice, so that over time, these were back-crossed for far more than 10 generations. Many of these strains were developed by Eva Eicher to study sexual differentiation and sex reversal effects. These have been back-crossed now with us many times over 50 or 60 generations to the black 6 background. The strains that were used represented a broad spectrum of inbred strains based on SRY allele polymorphisms coming from domesticus and musculus background strains. And those SRY allele polymorphisms were known to impact sex reversal in the context of a T. orleans mutation on chromosome 17. So in other words, depending on which SRY allele you had, you could have normal sex testes development, you could have what were called true hermaphrodites, so you would have ovotestes depending on the SRY allele, or you would have complete sex reversal in which these were male mice which had a reversal, which had ovaries instead of testes, all a function of which SRY allele I expressed. So we got these strains of mice to test whether or not natural polymorphic variation in the Y chromosome that was segregating amongst these strains influenced autoimmune disease. There are clear examples where mutations affect autoimmune disease or immune responses. One is YAA, for example. That was a radiation-induced translocation at Oak Ridge National Laboratories. This is an artificial induced mutation. And the mutation which Rhonda referred to in the 4 core genotypes is also a mutation, is a mutation which is not natural. It was a mutation that was found to occur in an embryonic stem cell line in which the Y chromosome was taken out of embryonic stem cell lines which had been multiply infected with retroviral over and over the years. So it's not a normal Y chromosome either. It has differences in terms of mutations that occur in embryonic stem cells in vivo. So we studied these mice. We studied them for 2 models, experimental allergic encephalomyelitis that Rhonda also discussed. And then another autoimmune model which we studied was Coxsackie virus B3-induced myocarditis. If you infect mice with Coxsackie virus B3 and then wait 3 days until day 7, take out the heart and study them for myocarditis. The immunization protocol we used in this case is called a 2X protocol. The animals get their MOG on day 0 and on day 7, and then we study them for clinical signs from day 10 to day 30. This does not use the ancillary agent of pertussis toxin that many people use. So here are the results that we published in our paper in Genome Research this past year. And if you just look at this is days post-infection, this is the mean clinical score, and these are the results of all of the different strains of chromosome substitution strains that we studied, with black 6 being right here at this point. It's clear that there's a continuous distribution of responses in terms of susceptibility or severity of the disease that one sees. And that is manifest is classically known as quantitative or polygenic inheritance, in which just simply replacing the Y chromosome on these 57 black 6 males can take you from a relatively resistant strain to a more susceptible strain. And we also saw the same effect in autoimmune myocarditis, in which these are all relative to black 6, the severity of the myocarditis autoimmunity in the heart. in which if you had 129, SGL, MA, PWD. So in both autoimmune disease models, the Y chromosome was able to modify significantly disease pathogenesis. So that was the first criteria of to test our hypothesis, is that the Y chromosome in fact influences autoimmune disease susceptibility in a normal setting. Nothing unique about these chromosomes other than that they're evolutionarily distinct. We then asked the question whether or not these SRY alleles and testosterone levels segregated with differential susceptibility. And here you can see the origin of the particular Y chromosomes. These are all musculus. These are mus domesticus A. These are mus domesticus B. These are broken down based on their ability to give normal testes development, true hermaphrodites, or complete sex reversal. So these are the alleles which bring about those effects in the sex reversal phenotypes. And as you can see, there's no clear distinction or correlation with any of the different origins of the SRY alleles in the phenotype that we saw. Since testosterone is a potential factor that might influence autoimmune disease susceptibility in this setting, as a function of SRY, the number of Leydig cells, for example, and testis and gonatogenesis, we took serum testosterone levels and asked whether or not there was any correlation there of the serum testosterone levels with severity disease using the cumulative disease score. And again, there was no correlation of testosterone development. In fact, the only real strain that had a huge, significantly different testosterone difference was RF. And it had very high levels of testosterone, and it's was a borderline in terms of its cumulative disease score. So basically, we couldn't find any correlation with SRY known alleles or with testosterone levels that correlated with with the effects of our Y chromosome on either EAE or myocarditis. So then, to test the hypothesis that Bernardo tested in Drosophila, was we set out and quantified the number of copy variants for 3 different multicopy gene families on the Y chromosome. RBYM, or RBMY, was one which was a ribosomal multicopy gene, very similar to Drosophila, SLY, and SSTY. And these are, again, these were there at the time of the sex chromosome evolutionary divergence, and have developed different multicopies in terms of on the X and multicopies in terms of on the Y. And some of those also have point mutations in them that just arise spontaneously. But for the most part, they're very different in terms of copy number amongst inbred strains or even wild drive strains. So we first looked at RBMY, and this is the cumulative. This again is the cumulative disease scores correlated against the copy number for RBMY, and there was a significant correlation of RBMY with susceptibility to EAE and susceptibility to myocarditis. We looked at SLY, and again there was a significant correlation of SLY copy numbers with susceptibility to EAE and a significant correlation with myocarditis. And as a control, we looked at SSTY, and there was no correlation of SSTY copy number with myocarditis or EAE susceptibility. So we in part filled the requirement of the Drosophila model in that copy number variation correlates with differences in disease severity and susceptibility amongst different males having the same identical genetic background. We then asked the question of whether or not these chromosomes could influence the gene transcription or the profile of CD4 T cells. So these are studies just taking naive CD4 T cells right out of the mice, no activation, they were not immunized, there was nothing done to them, because we believe that it is that basal state at which the environmental factors act upon to exacerbate or trigger autoimmune disease susceptibility. It's not some point of a cell that's been previously activated. So we think the basal gene expression profile is the critical profile that's important in setting up susceptibility to autoimmune disease once they see an environmental insult. And so we took the prototypical SGL chromosome and the B6 chromosome and used a false discovery rate of 0.05, and then we found 336 genes that were differentially expressed between these two, in CD4 T cells, between these two strains. And surprisingly, this is what almost blew us away. We were very surprised by this finding, that there are 2,859 genes that had different splice variants. So there was a huge effect, and of those, 398 exhibited differential expression of the alternate splice variant. So the Y, and the other point is that we looked at microRNAs and we saw no significant differences in microRNA expression in CD4 T cells as a function of the Y chromosome. So this polymorphic variation that exists in the Y chromosome is in some way affecting splicing of transcripts within CD4 T cells at the basal level before those cells are ever activated. Okay? Importantly, we went on to show that this translates into functionality. I didn't present this data, but it translates into functionality in terms of IL-17 production following anti-CD3, CD28 expression, gamma interferon production, several other cytokines. So just having that Y chromosome allele, SGL, in those CD4 T cells versus the B6Y chromosome gave very profound differences in IL-17 production, gamma interferon production without any activation prior to that. So there was a functional consequence to these alternate transcripts in some way, influencing cytokine production. We also wanted, because of this alternate splicing concept and the fact that, I'll go back, the fact that many of these genes, if we looked at Go terminology and categorized the differential expression, the processes that were primarily affected were metabolic processes, gene expression, regulation of signaling, chromosomal organization, and immune system processes. So this was very intriguing to us in terms of chromosomal organization and potential of the Y chromosome polymorphisms influencing epigenetically gene expression. So we wanted to look at the role of chromatin remodeling factors and see whether or not amongst those genes, whether there were Y chromosome was influencing chromosome remodeling genes. And in fact, so we ran a validation using a qPCR array that was specific for mouse epigenetic chromatin remodeling factors. And what you can see is the expression of B6 has very high levels of chromatin remodeling factors. And then when you have the SGL-Y chromosome in those CD4 T cells, you have a marked downregulation in chromatin remodeling factors. And that correlates with these differences in splice variant formation. So chromatin remodeling is impacted by Y chromosome polymorphism, which in turn regulates in some way the effector function of those CD4 T cells. And so that was our publication that came out in Genome Research sometime in 2013. So we wanted to turn our attention then and ask the question about, does the Y chromosome, as this natural occurring polymorphic variation, influence susceptibility to infectious diseases? And as I told you earlier, many species, males, including insects, are more susceptible to viral infections. Testosterone, as the sole mediator of the immunocompetence paradigm, fails to explain the ubiquity of the phenomenon. And so we examined the effects of the Y chromosome genetic variation in two viral models, Influenza A and Coxsackie-BV virus infection. So these again are just our panel of strains of mice. Well, first let me set the stage for why our thinking of this might be important. Saber talked about the sexual dimorphism in influenza, and we were very interested in trying to find out whether there was what the sexual dimorphism in influenza is pre-testosterone, okay, pre-pubertally. And in fact, if you go through the literature, it's very clear that young males before puberty are significantly more susceptible to influenza virus infection than are females. So if you go down here for 2009 H1N1 or seasonal influenza, you go down through these and look at these studies. Every single one, this is a sex ratio, male to female, 2.7, male to female, 1.8, male to female, 1.5, 1.3. If you look at confirmed H1N1, males to females are greater. Mortality, if you look at mortality, males are greater than females. And so every single parameter, except when you look at individuals who are 20 to 79 or older individuals, then is when you start to see this reversal in the sexual dimorphism. So this suggested to us, well, one more, this is a compilation of the incidence rates for seasonal influenza by sex from Japan from 2002 to 2005, and this is what happens as a function of age. So prior to puberty, 0 to 15 years of age, the sex ratio is male biased. At between this 15 to 60 years of age, it's virtually non-biased, and then when it's over 60, it's biased in the other direction. So there's a clear, taken together, these data suggested the role for non-gonadal hormones in the increased susceptibility of males to primary influenza infection or pediatric influenza infection. So we asked whether or not the Y chromosome influence could be responsible for that. So here are our results. These were all age-matched C57 Black 6 Y chromosome consomic strains. Our Black 6 control is right here. And so each one of these represent males with a different Y chromosome. And you can clearly see there's a significant difference. The p-value is highly significant when you compare them, the survival curves across these strains. And in fact, it breaks down into two clusters. Y chromosomes which don't have an effect on severity or susceptibility to survival versus strains which are highly significantly different to influenza infection. These, and again, I would point out here, there isn't a correlation here with autoimmune disease susceptibility. So the Y chromosomes don't correlate across immune phenotypes or disease phenotypes, and I think we have an explanation for that. So we asked whether or not, again, SRY alleles and testosterone correlated. Again, there was no correlation between SRY alleles and testosterone levels, so those aren't accounting for this difference we see in influenza. We then asked whether or not SRY, or the multi-gene copy number variation in our 3 multi-genes correlated, and clearly there was no difference, no correlation with RBMY, no correlation with SLY, no correlation with SSTY. So you can already see that Y chromosome, natural occurring Y chromosome variation influences immune diseases or infectious disease phenotypes, but it does it very selectively in terms of how it does it. We don't know what those mechanisms, what that means entirely, but it could be different copy number variations affect different phenotypes and in fact, different cell types. In fact, we showed in our paper in the genome research that the copy number variation that occurred in the BLAC6 as a function of Y chromosome impacted CD4 T cells, but when we looked at SJL mice, that copy number variation had no effect on CD4 T cells. It influenced gene expression in macrophages. Okay, so quickly then we just turn to the last one. We wanted to look at well, I'll just say that Coxsackie virus. Let me turn to this real quick. Coxsackie virus also had an effect. The Y chromosome had an effect on Coxsackie virus susceptibility, and here we saw that there was one correlation of Coxsackie virus susceptibility correlates with the copy number of SSTY. So in Coxsackie, SSTY correlates with survival. In influenza, none of them correlate with survival. In the autoimmune disease, it's RBMY and SLY copy number that correlates with survival. So back to this slide. This slide, we showed this also in EAE, that you have to think about sexual dimorphisms in an evolutionary context. You cannot think about them in isolation. And why I say that is based on this slide. These are all the results of plotting the genetic variation of our Y chromosome mice. Whoops, wrong one. Oh, damn. I gotta go back. Okay, these are all the results of plotting our male mice against B6 female mice. So if you take a B6 male and compare it to a B6 female, there is a slight sex dimorphism difference, as Sabra talked about. If you have a male with all of these Y chromosomes, that male, the sexual dimorphism is greater in males than it is to females. If you have a male with an AJY chromosome, there is no sexual dimorphism in influenza susceptibility between males and females. If you now have males which have SRY, BUB, PWD, or STY chromosomes, it reverses the sexual dimorphism and susceptibility to influenza virus. So everything in terms of sexual dimorphism, and we saw this with EAE, it's absolutely true with EAE as well. So sexual dimorphisms are evolutionarily contextual. And there's an interaction between sex chromosomes, X and Y sex chromosomes, that impacts the sexual dimorphism that one reads out. And presumably this could also occur in humans as a consequence of migration, because there are clear Y chromosome subgroups and interracial, or not interracial, but interhaplotypic marriages which bring into account different numbers of copy number variations across the X and Y chromosome. In fact, we think we can explain much of the sexual dimorphism that one sees in autoimmune diseases through this mechanism. We have a paper that's currently being reviewed in Science looking at the role of intragenomic competition between these X and Y genes. And in fact, it supports the concept, because if you look at SLE and MS, the number of birth sexes is very, very different across multiplex families where they're siblings. There is a sex skewing in favor of females. So autoimmune diseases in which there is a sexual dimorphism in the disease, there is also a sexual dimorphism in favor of the sex in the birth ratios. If you take type 1 diabetes, for example, which doesn't have a sexual dimorphism, there is no skewing in the birth ratio of those individuals across the multiplex families. So there is a clear connection between sexual development in terms of fetal outcome, survival, and susceptibility to autoimmune disease. And we think this can be accounted for by these multi-copy gene families and what's known as intergenomic competition. So I just will quickly move through this and just simply say, acknowledge my collaborators in the project. Our collaborators, Dr. Laura Case, Emma Wall, Julie Dragon, Naresh, who's now a postdoc in Mark's lab, Dimitri, Mohammed, James Zachary, Sally Huber, Elizabeth Blankenhorn, and the funding agents that support this work. Thank you.
So I've always thought your studies were so elegant where you do the consomics, where you take a given autosomal background and you put in the Y gene for one strain versus another strain versus another strain to show some strain-related allelic differences can have an effect. First of all, I don't think that's necessarily mutually exclusive of an effect of maternal imprinting on X genes. But I would also say what we've had to do with Art Arnold is I wonder when you measured those testosterone levels, because you know, testosterone levels could be different very early, during thymic training, and they could have effects on the immune system. And we had very much difficulty, and we actually showed that our mice that were for sex chromosome differences, they were not hormonally different, because we had to go back to things like looking at anogenital distances and areas in the CNS that were amenable to sex hormone differences on their structure to actually prove to people that, you know, you just can't measure testosterone at one day and say that the SRY allele wasn't different, that it all could be explained by differences in testosterone. So,
well, SRY alleles,
yeah,
that's very clear. There's no
so did you measure testosterone, you know?
We measured testosterone in age-matched males. Oh, we
Not
measured.
how many different time points, I guess, is the question.
Yeah, we did one time point at the same time of day in age-matched males. So we presumably that we have a snapshot window.
Right, so they were like
which
4 weeks
says,
old, or how old were they?
I don't remember. We have to go back to the paper, but they were post-pubertal, 6 weeks
So
old.
my point is they could have been different developmentally. Those different alleles of Y could have had different testosterone levels at day 3, at day 5, at day 7, at 2 weeks, 4 weeks of their life.
Oh, I'm not arguing.
Yeah.
I'm not arguing with that, but I'm just simply
Yeah. saying
at the time that you look at the transcriptomes of
Right.
those mice, there's no effect of testosterone
At that
on
time
that point in
Yeah.
time. I mean,
Right.
same with the SRY deletion.
That's why we
you
went
don't
back
look.
to
You
ask.
haven't looked at.
But that's why we didn't just look at a few time points. We actually looked at structures that were known to be influenced by differences in sex hormones to ask whether they were different in the SRY mice.
Yeah, we've looked at anogenital distances, and that we can't see any anogenital differences in any system, whether you give them BPA or any estrogenizer. It's a very bizarre assay. We could not see any differences, so we filled that criteria as well. So we didn't see any differences in anogenital distances with our Y chromosomes. Yeah.
That's good. Mm-hmm.
Yeah.
So, what were the ages of your mice for your for your flu work? Just
Right,
were
they're
they
all-
pre-pubertal?
No, they're adults, male.
And so, what happens? Just because it is, and I love that you hit on that increased susceptibility of younger, pre-pubertal
Mhm.
boys relative to girls. So, what happens if you use pre-pubertal?
We haven't. I don't have any money. I can't get money to. If I could get money to do the experiments, we'd look at pre-pubertal males as well.
Do you think the effect would be greater prior
I
to
think
any
it-
effect
yeah.
of sex hormones?
Yeah, you may see a greater impact of a role of the Y chromosome polymorphism in those males.
Yeah, prior to that spike.
Prior to the testosterone surge
Yeah.
with puberty. Yeah.
Okay.